Good day, everyone, and welcome to our Cell and Gene Therapy workshop. My name is Emma, and on behalf of Oxford Global, I'd like to thank you all for joining us today. Today, we have three fantastic presentations focusing on different aspects of improving quality and yield for viral vector manufacturing using osmolality. Our speakers in order of appearance will be Kendall Studd, Scientific Applications Specialist at Advanced Instruments, Adrian Thula, Process Development Scientist at the Cell and Gene Therapy Catapult, and Lucia Marani, Analytical Development Scientist at Oxford Biomedica. Following the three presentations, our speakers will then participate in a panel discussion to answer your questions. Please submit your questions through the chat option on the bottom of your menu display. If you would like a specific speaker to address your question, please ensure to include their name in your submission. There will also be polls available to answer following each talk. Please do take a few moments to fill these out. Finally, the full recording of the webinar will be made available to download on our secured website, and the details will be sent to you after the webinar. Without any more delays, I'd like to get started with our first presentation. Kendall, over to you.
Hi, I'm Kendall Studd, Scientific Applications Specialist for Advanced Instruments. I just wanted to say thank you for joining this workshop today, looking at the use of osmolality to improve yield and quality within viral vector production and manufacturing. Advanced Instruments are extremely focused on cell and gene therapy as a whole right now, and we aim to prove today how the incorporation of this critical process parameter can potentially significantly improve your bioprocesses. On today's agenda, we'll be focusing on osmolality as a key parameter within all sections of the cell and gene therapy workflow, from upstream and downstream to then your fill, finish and final product. Included in that, I'll be taking a look into retro and lentiviruses in particular also. And then to conclude, I'll be presenting what AI can provide to you as a customer, looking to include osmolality testing into your laboratories. We'll also be putting some questions to the audience via the chat box throughout the workshop, and we would really appreciate it if we could get as much feedback as possible, as this in turn will help us help you. To start things off, although most of you will be probably aware, osmolality is defined as the concentration of particles or solutes dissolved in a solution, expressed as osmoles of solute particles per kilogram of solvent, or milliosmoles. Solute concentration in process fluids is an important parameter required in bioprocessing to achieve consistent results. Osmolality is a measure of concentration and is therefore considered a critical quality attribute and critical process parameter in bioprocessing. Due to the measurement being dependent on mass and not volume, it is not affected by a change in the solution's temperature. Here we have a generalized overview of the cell and gene therapy workflow, with the left-hand side representing your upstream processes and the right-hand side representing your downstream processing. Osmolality really can provide a flexible parameter, giving you a robust analytical tool to help maintain that critical control over your operations. Let's get started with upstream processing applications, where you want to be optimizing those culture and media conditions, maximizing your cell health and density, and ultimately your ideal end goal of increased viral yield. As you can see in orange here, we're first of all going to be looking at media quality control. Media QC is a commonly known use case for osmolality. However, there is now evidence of a growing trend in biopharma of outsourcing media from external vendors. This calls for heightened QC for these off-the-shelf media and similar testing for in-house manufacturing. Cells need a physiologically relevant environment, so osmolality is a key parameter. There is clear evidence that falling out of specification affects the growth rate and productivity of HEC293 cells, obviously a key cell line for a lot of viral vectors. This can then be compared to the more stable nature of monoclonal antibodies, which are less susceptible to environmental changes. Obviously, once you have your chosen cell line and your media within the correct specification, you need to be able to optimize your conditions for that cell culture expansion. First off, I wanted to show the usual use case for osmolality within cell culture monitoring. The key to the use of osmolality is that of optimization. You can incorporate osmolality testing to have that in-process check for optimal cell growth and health, as well as a check for any potential contamination, which could in turn lend itself to a batch deviation and therefore batch failure. In summary, osmolality is capable of telling you whether your cells are happy, if they're being productive, with the potential to increase this, as well as the use of osmolality to potentially reduce your risk of aggregation. So how exactly does this work? If you take stem cells as an example here, osmolality has been seen as an important process parameter which has the ability to preserve cell viability and productivity. Various parameters are also obviously going to come into play here, such as pH control or various feeding strategies, but these all will in turn impact the osmolality within your cultivation conditions. You are also wanting to keep that osmolality within your set specification. An extreme osmolality within your media, for example, can compromise your cell growth rate of your stem cells. However, it has been shown that a higher osmolality can actually be used to slow cell growth to then maximize your productivity. The information shown here is somewhat incorporated into the collaboration advanced instruments have had with Cell and Gene Therapy Catapult. We are so excited for Adrian to present to you this incredible new data which has so much potential to change your upstream workflows and improve and increase not only yield but quality too. Moving on to one of the final upstream use cases, that of vector stability. Stability is a huge critical quality attribute for any product. For viral vectors, this attribute is perhaps more critical because of the need to preserve stability throughout processing. Here, you can see some highlights from key publications about retroviral vector stability. At the top left here, the literature showed that higher media osmolality contributed to improved vector stability and cell productivity. Why? Well, for one, higher osmolality has been associated with a lower cholesterol to phospholipid ratio, contributing to higher stability, which was shown in the second publication on the right here. Another factor is sugar metabolism. Increased levels of sorbitol positively correlates to a retroviral stability and productivity. I'm going to talk in more detail about a couple of these papers shortly and show how this research can then provide evidence and data into the furthering of work within lentiviral vectors and the inclusion of osmolality testing. Before we jump straight into the publications though, I wanted to highlight what knowledge is currently out there surrounding osmolality and retroviruses, and then in turn lentiviruses. So lentiviruses may not be the most commonly used viral vector out there, however the numbers of companies researching and developing is growing, and we see a real interest and curiosity around this vector workspace. You will note that we also have Lucia Morani presenting later in the workshop, who is able to give us a real insight into how osmolality testing is incorporated into Oxford Biomedica's lentiviral work. So, there is current evidence to show that osmolality influences the overall stability for retroviruses. And there is also evidence that lentivirus stability can lend itself to a decreased risk of aggregation. So a logical conclusion would be to say that if osmolality can influence stability in retroviruses, it would also be relatable to the lenti stability and ability to aggregate. Moreover, it is also known that osmolality can influence the productivity in both retroviruses and the subset of lentiviruses too. And I hope to support these statements and hypotheses with my next few slides. This is one of the publications we saw earlier. It is by Anna Corradina and it is surrounding vector stability. Looking into this paper in more depth, it details how osmotic pressure can play a role within vector stability, focusing on retroviral vectors. I've included the abstract to the article here, and I wanted to highlight what I think are the key parts which can be taken away from the literature. It first highlights how one bottleneck or pain point in the achievement of high titers of retroviral vectors is simply down to the instability of the vector itself. Within the research, the cell culture media was raised from 335 milliosmoles to 500 milliosmoles, using either ionic or non-ionic osmotic agents. What was seen was that the infectious vector inactivation rate was inversely correlated with the osmolality of the production media, therefore concluding that the use of these higher osmolality medias actually enhanced vector stability. It was also seen that with the addition of sorbitol to increase the osmolality, the production rate also increased. The last significant section of the data showed that the smaller percentage of cholesterol to phospholipid ratio in the membrane the more stable the viral vector. To summarise what this data found, it can be said that the use of osmolality testing to manipulate that increase in cell culture media increased the stability of the viral vector, and therefore a higher yield was achieved within production of the retrovirus. Moving on to the next publication, again Anna Corradini was involved here, which looked at both retroviruses and lentiviruses and the challenges scientists are seeing today. The data shown here also supports the idea that the higher the osmolality, this time increased with the addition of either glucose or fructose, the increased cell productivity. They also took note of the cholesterol to phospholipid ratio, with this all again suggesting that when having various parameters and additions to your media, the more likely you are to improve your viral stability and productivity. If this is the case for retroviruses in particular, why can it not be the case for lentiviral vectors too? We've talked through various upstream applications within cell and gene therapy. Now we can look into the downstream ones, where we should be checking that raw material concentration and performing in-process monitoring to maximise purity, stability and yield. Buffer quality control can be comparable to that of your media QC use case within your upstream processing. However, we do have some more in-depth data for this specific application. I mentioned earlier that osmolality is independent of temperature, making it a great parameter for bioprocessing. As we move into the downstream uses, I want to visit some data from an investigation completed in 2019. Across a wide range of buffer and buffer component concentrations, we see that osmolality provides a consistent and predictable measurement, specifically when comparing that to conductivity measurements, which are shown here in this second row of graphs. While not perfectly linear, these curves in the top row show how osmolality is a great orthogonal tool to not only the conductivity measurements, but pH also. This became apparent during a recent purification study by Professor Alois Jungbauer and team from the Boku Institute in Vienna, which showed osmolality's importance as a predictive measure of protein concentration. Although the paper doesn't focus on proteins, we do see real relevance within the cell and gene therapy workflow, and hope that the paper and the data shown here can be seen as a starting point for a discussion around the significance of osmolality testing within downstream processing. Moving further down the workflow into your filtration operations, osmolality can be used to verify the completion or quality of concentration within your ultrafiltration diafiltration steps. As well as verifying completion of buffer exchange, it has been suggested that osmolality can also provide information about potentially reducing the number of diavolumes within your filtration operations. If this is of interest to you, please get in touch and we can go into a little more detail regarding this specific use case. Additionally, we are looking for insight or a collaboration which can look at osmolality as a potential parameter used to check and minimize vector aggregation to generate more data to support this publication listed. The specific study here looked at using osmolality to check the levels of different excipients, such as magnesium sulfate, sodium chloride, arginine and lysine to name a few, which can all prevent aggregation of AAV2 vectors. To focus in more on the use of osmolality to confirm the completion of buffer exchange, I have included two examples and also this bar chart to show how it could exactly help. Example 1 is the use of osmolality to simply check the efficiency of buffer exchange by measuring your permeate, whether that be a single measurement, a duplicate, a triplicate, all dependent on your lab's SOP. Example 2 is related to the chart, showing how you can assess the optimal number of diavolumes needed within your filtration operations to then potentially minimize this process if you are able to confirm you have reached your osmolality specification previous to your maximum number of diavolumes. So what we are basically saying is that by monitoring your osmolality trend, you can determine when that plateau has occurred and can therefore reduce the number of diavolumes necessary, in turn saving you time, money and resources within this section of your workflow. We were lucky enough to have Kevin Lin, formulation scientist at Alexion, present for us last year, and he was able to discuss this osmolality use case in more detail within his experience at Alexion. If you would like to know more, we can share this video with you to watch at a later date. And finally, looking at the fill and finish section of this workflow. Osmolality testing has long been a part of the final product release testing for therapeutics. But what are some of the other applications for osmolality within this workflow segment? I want to really focus on cryopreservation and how osmolality comes into play. Cryopreservation is meant to preserve the integrity of drug substances and their CQAs, and therefore maintain that high rate of recovery of cells post-thaw. By testing the osmolality of the cryopreserves before freezing, you can ensure the composition is correct, providing confidence in the upcoming preservation steps. By doing this, you are potentially saving you and your team time, money and effort, as you will be able to pick up on any batch deviations before it may be too late. As you all may be aware, some of the current vaccines for COVID-19 need to be stored at an ultra-low temperature to maintain integrity. This can be seen as a significant issue throughout the shipment process. However, cryopreservation may be one solution to this. As you can see from the graphic, osmolality can be applied at various times throughout the workflow, with big potential for those shipment phases. We do get questions about using freezing point depression osmometers to monitor cryopreservatives that contain ingredients like DMSO and PEG. So, can a freezing point depression osmometer actually test samples which include anti-freezing agents in their composition? This data from a recently completed study showed that our freezing point depression osmometers can successfully and precisely measure the osmolalities of these samples, looking at both commercially available and homemade solutions. Cryopreservative formulations are known to have a higher osmolality range, as can be seen here, with these specific solutions starting from over 1,000 milliosmoles, and even at that higher end of the 2,000 mark, the osmometer showed sound reliability and repeatability for all recipes tested, supported by the standard deviation and coefficient of variation results. We can now look at the downstream processes for lentiviruses in particular. It is known that lentiviruses are produced with the help of serum, which ideally assists in that optimization of cell growth and vector stability. Albumin can also play a role within vector stability when looking more at the downstream processing side of things. However, with the drive now to reduce the use of serum as they are bovine-derived, what is it that can be put in place to help improve that vector stability and for the optimization of cell environment? Of course, osmolality would be my answer here. If you incorporate osmolality testing within your process, whether that be upstream, downstream or both, you are adding that extra check or process parameter to your process, keeping tabs on those critical quality attributes that are so key to maintaining an efficient workflow. This can then be brought back to the work done by Anna Konradina. If you have that robust controlled process, you are producing more stable vectors and therefore are increasing your chances of a higher yield. I previously mentioned aggregation in one of my first slides, but here is a small extract from a website which states very directly that when including a diafiltration step to your filtration operations, it is the perfect opportunity to control your potential virus-to-virus interactions and therefore reduce the risk of aggregation previous to that sterile filtration step. If you want to optimize that step even further, it is suggested that you should not only be looking at the ionic strength and pH, both of which are obviously usual practice, you should also be looking at your osmolality of that formulation buffer. So here we have a reviewed workflow. And by having that extra control over your process with the inclusion of osmolality testing, you are increasing your viral stability, reducing your chance of aggregation, and also increasing your yield. So why would you not want three things at the expense of a 90-second osmolality test? And with that, that concludes the run-through of osmolality applications within the cell and gene therapy workflow. I hope I was able to increase your understanding of osmolality within the more commonly known use cases, as well as bringing to your attention some potentially newer ones. We can now look specifically at what advanced instruments can provide to help. At the basic level, we can offer robust osmometers for low and high throughput workflows. Our Osmotech, as seen on the top here, is a single sample osmometer developed specifically for biotech customers. We also have its multi-sample counterpart, the Osmotech Pro, underneath, as well as our newest single sample edition, the Osmotech XT, that was launched September 2020. This specific instrument incorporates an intelligent freezing technology, allowing for the measurement of higher osmolalities up to 4,000 milliosmoles, perfect for those viscous, highly concentrated complex samples. These instruments provide the obvious osmolality testing with high accuracy and precision. But we've also incorporated many features that have been deemed important by our biotech customers. All instruments are easy to use with efficient test times and small sample sizes. There are significant data management and connectivity features which make for secure and efficient management of electronic data. There is 21 CFR Part 11 compliance, and another new feature launched with the XT is the second electronic signature for that review and approval of data. All features here are really giving you that full compliance with all Alcoa principles and data integrity regulations, whether that be within the expected GMP environment or even within your R&D and less regulated areas, which are now seeing an increase in need for these integrity features. And like I've already mentioned, the higher range for the XT, giving you the ability to accurately and precisely measure previously difficult samples. I also just want to make you aware of a new instrument we are currently working on, a new technological development including a 96-well plate-based osmometer, which will be able to meet those demands we are seeing within upstream cell culture development. It will be targeted at users of the AMBER and other high-throughput automated systems. So all that is left to say is to watch this space. We recognise that a growing field with ever-changing regulations requires flexible devices. You can see here the data connectivity options of the latest instruments, which vary from the ability to connect to a LIMS or a LIS, to being able to network share, and also using a remote web server to access your data. All these options allow for the easy integration of the osmometers into any work environment, especially that of a GMP-compliant lab. We have developed a thorough portfolio of support options for customers, from installation and training to IQ/OQ validation packages, which then specifically allow for that easy installation and qualification of your new device, without the need to develop your own procedure, saving you both time and money. We also offer service packages that include preventative maintenance and technical support when needed. Thank you for listening today, and I really hope I have shown you the flexibility of osmolality testing and the osmometers that can help you within bioprocessing. On the right-hand side here, we have a document we at AI have produced looking into all of the applications for osmolality within the production and manufacturing of cell and gene therapy products, which is available on our website. Here is the link you'll need to access this and many other documents, white papers and application notes that are available on our website. If you want to contact me further, please don't hesitate to drop me an email at the address shown. Otherwise, I'm active on LinkedIn and will get back to you as soon as possible. One more question from me today. So we know that regulatory guidance within these sectors are at some point unclear and are ever-changing and ever-evolving, but we would love to hear your thoughts on this topic. Please see the chat box for the question and let us know your experience surrounding this common discussion point in cell and gene therapy. Next up on the agenda, we have Adrian from Cell and Gene Therapy Catapult, who is going to present this brand new data we are all so excited to bring to the world of pharma. We know there are 3 common pain points within viral vector production and manufacturing: the yield, the quality, and the workflow efficiency, and we hope that with this new data we can improve all 3. Again, thank you for your attendance today, and I hope you enjoy the rest of the workshop.
Thank you very much for that great presentation, Kendall. Attendees, you should now be able to see our first poll on your screen. This will be up for the next few moments. Please do go ahead and add your responses now. Fantastic, thank you so much for responding. We can see that it seems to be quite an even split between all 3 options, with only a few of you outsourcing as only measured in QC. Our next poll should be available now. Fantastic, thank you again for your quick responses. I can see the majority of you aren't sure, and then the next popular answer is audit trail. That's great. Our speakers will comment on these during the Q&A following the presentations. Next up, we have Adrian Sula presenting. He is Process Development Scientist at the Cell and Gene Therapy Catapult. Adrian, thank you for joining us. Over to you.
So my name is Adrian, and today I have a presentation of the impact manufacturing. So I will show you today part of a collaboration between Advanced Instrument and Cell and Gene Therapy Catapult. So this is because there's still a lot of challenges on AAV manufacturing. So we can still see some low process yields. Some unit operations are still not optimal. And of course, a lot of people know that the costs are still relatively high. So here on this slide, you can see a generic process for AAV manufacturing, where you start with your cell expansion, you then go into a stir tank bioreactor for production. And then finally, for the upstream, you have to disrupt the cells in order to release your AAV. So you can do that with a chemical lysis, for example. And then you go to the downstream, where you can do clarification, then followed by capture, polish, ultrafiltration, and diafiltration. So today, I'm going to focus mainly on the upstream, but I will also talk about the impact it can have on both upstream and downstream. So here at Catapult, we have different platforms for different purposes. So we can do some screening and DOEs at a very small scale, so 15 ml. Then we can do some optimization at 250 ml with a scaled-down model. And finally, we can scale up to 2 liter or 50 liter. And the data I'm going to present have been generated at small scale, so 15 ml and 250 ml scale. So the process parameter used to generate those data are the following. So we are using HEC-293 suspension cells. We are doing a transient transfection with AAV1 serotype. And the media we use for this experiment is balanced CD, which has a pH between 6.7 and 7.4 and an osmolarity range between 280 to 320 milliosmol. Concerning the analytics of the experiment, so we are monitoring osmolality thanks to the Osmotech Pro from Advanced Instrument. We are also monitoring cell counts and viability, metabolite. We are assessing transfection efficiency 1 day post-transfection thanks to flow cytometry. And we are analyzing Titer thanks to qPCR and ELISA for viral genome and total particle. So today, I have 2 data sets from 2 different experiments. So I'm going to start with the first one. And during this first experiment that happened at small scale, 15 ml, we went to have a look at the impact of shifting osmolality during the AAV manufacturing process. So we also wanted to have a look at the impact of using 2 ways of shifting osmolality. So using an ionic way or non-ionic way. So you can see on the slide all the conditions we use for this experiment. And basically, we fixed osmolarity at 300 milliosmol for transfection. And we did a shift at 2 different time points, 24 hours post-transfection and 48 hours post-transfection. We did this shift at 2 different set points, 400 milliosmol and 500 milliosmol. And we did exactly the same thing with both components. So sodium chloride for shifting osmolarity with an ionic way, and using sorbitol to shift osmolarity with a non-ionic way. So before going into all the results, just as a start for the experiment, so the transfection efficiency that is determined a day post-transfection was assessed before any shift in osmolarity. So we were expecting to have a similar level for all the conditions. And you can see it's around 40% transfection efficiency for all of them. So this was as expected. It's a starting point for this experiment. Before showing you all the titer, I just want to show you some trends we want to identify from the result. And I'm going to start with the difference between using a low osmolarity and a high osmolarity in the process. So what you can see the difference between condition from 300 milliosmol in red, 400 in green and 500 in gray. And we can see a trend for a decrease in variability when going into high osmolarity and a decrease in cell diameter. By doing a comparison between ionic, non-ionic, and the control with no shift, what we can see is so sodium chloride in red on the bottom graph, sorbitol in green, and the control in gray. What we can see is a trend for this lower variability and a decrease in cell diameter when we use sorbitol compared to sodium chloride and the control. And I'm not showing the data on this graph, but concerning the metabolite level for all those conditions, all those comparisons, they were similar for all conditions. But having a look at the difference between shifting osmolarity at 24 hours post-transfection, at 48 post-transfection, and no shift, what we can see is like there is a trend for a decrease in lactate and ammonia accumulation when we do the shift later in the process, so at 48 hours post-transfection. So now I can show you some results about the titer, which will be of interest for most of you. So what you can see on this slide is the titer on the left for all the conditions. On the right, what you can see is a specific titer for all the conditions again. And what we saw was a trend for an increase in titer when we were using a shift with sodium chloride at high osmolarity, so 500 milliosmol, compared to the use of sorbitol. We also see a trend for an increase in specific titer when using the same condition, a shift with sodium chloride at 500 milliosmol compared to the control. And our conclusion for that was there was a significant increase in titer for a shift happening at 48 hours post-transfection at 500 milliosmol using sodium chloride. This compared to our control condition. So one more thing, what you can see at the bottom of this slide is we try to see if there was a trend with osmolarity at lysis compared to the titer. So we didn't see any trend between the titer we got and osmolarity at lysis and the specific titer. But if we only have a look at sodium chloride, we can see a trend for an increase in specific titer compared to the osmolarity at lysis. And what you can see on this slide, on the top, it's the conditions that had a shift at 24 hours post-transfection, and at the bottom, the conditions that had a shift at 48 hours post-transfection. And we can see a trend for an increase in specific titer when increasing osmolality using sodium chloride. We could not see any correlation for sorbitol, and we could not see any correlation with titer. It was only for specific titer. So for this experiment, finally, I want to show you something about empty full ratio, because it's something we would like to increase in AAV manufacturing, increasing empty full ratio. We want more full, obviously. So what you can see is at the left graph, the total particle for all conditions. On the middle one, you can see on the middle graph, what you can see is the specific total particle titer. And you can see a trend for a decrease in total particle when we shift osmolality later in the process, so at 48 compared to 24 hours. And if we combine those data with the previous data, what is interesting is we can see a trend for an increase in empty full ratio when we shift osmolality later in the process at 48 hours. And this is what you can see on the graph on the right. So you can see an increase from something like 10% to almost 20% in certain conditions. So after this experiment, which was a screening and very exploratory, we did an experiment, and this will be my second data set, an experiment at 250 ml scale. So this experiment was 6 vessels and 3 different conditions, so condition in duplicate. And those 3 conditions were the following. So a control condition with our process where we control osmolality at 300 milliosmol during the process. A second condition when we decided to shift osmolality at 500 milliosmol, 48 hours post-transfection. And finally, a last condition where the same thing, but a shift at 72 hours post-transfection. And we achieve this osmolality shift thanks to sodium chloride. And it was a feeding on the bioreactor. So it wasn't a bolus addition, it was a slow feeding of roughly 30 minutes. So during this condition, during this experiment, we wanted to basically have a look at the distribution of our AAV between the cell and the supernatant. So that's why we decided to go for lysis at 3 different time points to assess this distribution. And we did it at 48 hours post-transfection, 48 hours post-transfection, 72 hours, and 96 hours. So for every time point, we did 3 different samples. So first, a standard chemical lysis of the cell culture. Then just a supernatant harvest treated with nuclease. And finally, only a lysis of the cell after being spun down. And this was in order to assess overall productivity for the lysate, release of AAV for the supernatant, and intracellular AAV for the lysed pellet. And one thing to know about this experiment is we decided to extend the production period. So, and we also decided to not use any PID. So this was in order to not affect osmolality and really having a look of at the impact of osmolality on our process. So because of that, we have quite poor variabilities, especially at the end of the process. And we also had a slight deviation of one of our controls. So one of our controls had a lower cell density from the beginning of the experiment. And you will see like our control has some variability, but this can be explained because of this deviation. So concerning the titer, what you can see on this slide is from the left to the right, the 3 different time points. So 48 hours harvest, 72 hours, and 96 hours on the right. And what you can see is on the top, the 3 graphs on the top are viral particle, a specific viral particle, and on the bottom, a specific viral genome. So what is interesting is, so you can see on those graphs and the following slide, the same color code. So on in blue, it will be the control. In red, it will be the conditions that had a shift at 48 hours post-transfection. And in purple, the conditions that had a shift at 72 hours post-transfection. So on those graphs, what you can see is supernatant versus lysate. And we can see, it's quite obvious that we have more extracellular when we do a shift in osmolarity, both for the shift at 24 hours and 72 hours. And we can also see a trend for highest viral genome and viral particles, so specific viral genome and specific viral particle. So what is, I think, the most interesting is if we calculate the percentage of extracellular viral genome, this is the same color code, so for all the three conditions and the three different time points. And you can see at 96 hours, it seems that we can maximize extracellular viral genome when we have a look at the supernatant versus the overall productivity of our condition. So we are at 94, 93 hours for both conditions who had a shift in osmolality, compared to 70% for the control condition. So the variability in the control is because of the deviation I talked about. But interestingly, if we have a look at the same thing for viral particle, we have the same trend. So this time it's slightly lower in terms of extracellular viral particle. We have 84, 82% for both conditions who had a shift, compared to 61% for the control condition. But if we have a look as well at empty full ratio, what is interesting is we can see a trend for an increase in empty full ratio, having a look at empty full ratio of AAV in the supernatant. So we go to 23, 25% for condition with a shift, compared to 20% for the control condition. So all of that is still early result and quite exploratory. But we can think about the potential impact it can have on our AAV process. And basically, like we saw, like we could increase the yield of the production, and by itself, it's already like a big win, because you're gonna decrease the cost of your process. But we could also think about removing the cell disruption step, because if we get rid of the lysis, we're gonna decrease the cost. We could basically think about maximizing AAV release in the supernatant, do supervest directly. And again, it's a decrease of the cost, but we could even like think further and get rid of the clarification, do direct capture. And in this case, we use the DSP, we reduce the time, and we reduce the cost again. So all of that is potential impact. Of course, doing some cost of goods modeling would be the best. But thinking about the solution, we only use sodium chloride in order to control the osmolality, which is cheap, an easy solution to implement. It's only a feeding. And it's also like scalable if you want to scale it to manufacturing. So thank you for listening, and if you have any question.
Thank you very much, Adrian, for that fantastic presentation. Up next, we have two more polls for you, which should be visible now. So the first one is, what are the main challenges in your viral vector process? Fantastic. Thank you so much for providing these answers. And I can see that overwhelmingly, it seems that low or inconsistent yields are one of the most significant challenges in this process strategy. We do have a second poll following Adrian's presentation, which should be available now. Great, thank you very much, everyone. And it looks like the overwhelming answer here seems to be that most people would not consider removing the lysis step currently. So we'll now move on to our final presentation, which is from Lucia Marani, Analytical Development Scientist at Oxford Biomedica. Lucia, thank you very much for joining us, and over to you.
Good afternoon, and thank you everyone for joining at the webinar today. My name is Lucia Marani, and I work as an Analytical Development Scientist at Oxford Biomedica. I'm sure you're all aware of what osmolality is and how it can be used in both the healthcare sector as well as the biopharmaceutical one. And of course, in particular today, I will be focusing on how we can use it at Oxford Biomedica for characterizing our lentiviral vectors, as well as improving the manufacturing process of them. So this is just a schematic representation of what the production process looks like for our vectors, and it can be divided into upstream processes as well as downstream processes. And so in particular, we could, for example, optimize the cell culture media, or on a later stage, find a way to use osmolality to prevent vector aggregation, increase titer, and of course, even changing the buffer formulation to better suit the end application. I will be talking about different stages of the manufacturing process and different samples, but for most of these samples, I will be focusing on the very last of the stages. So I will start by showing you how we used Osmotech single sample microosmometer to develop a method for osmolality testing of lentiviral vectors. Of course, we started by performing calibration and verification of the instrument, and then we proceeded by measuring different lentiviral vector samples in order to gain an understanding of what the expected values for these samples were. And then we decided to question whether the method could be used as a stability indicating method by inducing force degradation. First by performing freeze-thaw cycles on these samples, and then by storing the samples at 37 degrees. So the recalibration matched the expected value ranges that we wanted, and of course we then selected certain standards to be within the recommended plus or minus 50 milliosmolar range from these expected values in compliance with the US and European pharmacopoeia. And then we decided to also observe the accuracy of the measurement by performing triplicate measurements rather than just a single measurement for the samples. And of course, this led to the definition of a standard procedure and a schedule for the maintenance and calibration of the instrument. So I'll start by showing you how different manufacturing stages behaved in terms of osmolality. As you can see, there were different values obtained, and this was done as a methods precision assessment as well over 3 different days and by 2 different operators. There is a slightly higher variation when we look at one of these stages. However, this may be due to the intermediate precision measurement being slightly higher in terms of mean values compared to the previously obtained one. And at this point, it wasn't necessarily a matter of concern. It was just an initial characterization that was performed. And then we decided to perform the freeze-thaw cycles on the same samples. And this was done by thawing the samples from the normal temperature, which is minus 80, and then measuring for the first time after storing them again at minus 80 to measure a second time, and then doing the same thing for a third cycle. The pattern of the osmolality levels is quite similar, as you can notice, and just to help with that, you can see that these were the previously obtained results. So the control versus freeze-thaw cycles mean values are actually very comparable, and this was good news. However, we needed to see whether we could use this method as a stability indicating method, and therefore it was essential to look at more in depth how we could force these samples to actually degrade and see if osmolality would change after that. So to do this, we store the samples at room temperature for 96 hours and compare to an incubation at 37 degrees. And this was done obtaining really not really exciting results, in the sense that there wasn't much of a difference when the samples were stored in control conditions or room temperature conditions compared to 37. So it was necessary to extend this degradation and also to, in that case, to use as control samples that were stored at minus 80 degrees, which is indeed the standard storage temperature. And this was done up to day 11. So as you can see, there is an increase in variation after time point 6, and this variation is also showing not only increased variation, but also higher values of osmolality, mean values of osmolality overall. there wasn't a huge difference at the end in terms of mean values, but you can see that there is indeed a higher variation when the samples are kept at 37 degrees. And this was also observed for other samples. As you can see here, we have another example, and this was again comparable to two other samples in the same kind of formulation buffer. The overall mean values are very comparable from control to 37. However, there is a slightly higher variation when the samples are kept at 37 degrees. So this is probably suggesting that there is something happening, but we're still not seeing anything major in terms of changes or changes in mean values overall at the end of the experiment. Until we obtain results from this sample specifically, in which the storage at 37 degrees caused it not to be homogeneous anymore. And of course, having a homogeneous solution before measuring osmolality is essential. And that's why these values are marked in red, is because they were above the accepted value for variation that we set in the method. And probably a little bit different from previous results. This time, the overall mean values for osmolality were lower rather than higher or similar to control. This is not necessarily negative or positive. It has to be taken as it is because, of course, this is due to the sample not being homogeneous anymore, and therefore the measurement cannot be accurate enough to give us a certain value. So to summarize, we observed how different manufacturing stages and different samples will require the use of different standards for the daily verification, and there was no impact in osmolality values when the samples were freshly thawed and measured compared to after they underwent 3 cycles of freezing and thawing. There is a noticeable increase in variation after the samples were forced in degradation after 7 or 8 days at 37 degrees. And also this showed higher variability and loss of accuracy over time, especially for one sample in particular. So to conclude, the method was set up as a GMP method in the company, and it is now part of the stability indicating assay portfolio run at OXB. There is, of course, great interest in integrating the osmolality testing also in other parts of the manufacturing process to improve the both the upstream and downstream processes for lentiviral vector production. And these last points was about testing different formulation buffers as controls in the forced degradation conditions to try and understand whether the lack of a homogeneous sample was due to the buffer itself or other components within the actual product. And with this, I would like to thank you everyone for your kind attention. And of course, I'm very happy to take any questions, whether now in the chat or feel free to contact me via email afterwards.
Thank you very much, Lucia, for that great presentation. We now have one final poll for you all to answer, and then we'll get straight into our panel discussion. Great, thank you very much for all of those quick responses to this final poll, which was, what are the main limitations of your current test for the stability of viral vectors? And it looks like the result overwhelmingly was that you outsource your vector stability testing. So we'll get straight into the panel discussion now. Our speakers can view all audience questions that have been sent through, and we'll read them out before providing their responses. Please do continue to send questions through using the chat feature on the platform, and the panelists will be able to incorporate these into their discussion. So thank you very much, Adrian, Kendall, and Lucia. Over to you.
Thanks, Emma. I think the first question that's just come through I'm going to take. So the question is, how far off is an inline osmolality test? So we actually get this question very commonly at advanced instruments, and a lot of our osmometers are used at line, but in terms of inline or online sensors, it is something we're looking into, and we are really focused on getting an instrument out there. However, unfortunately, we don't really have a specific date or a timeline to give you. But yes, it is something we're working on, and we hope to bring this out in the future at some point. So thank you for that question. Okay, so I think there's a second question that's come in for myself. I'm not sure about Adrian and Lucia, but a question came in regarding cryopreservative formulations and how do our osmometers actually measure these more viscous samples with components such as high PEG and or mRNA. So first of all, going back to my presentation, which looked at the Osmotec XT, this instrument actually is using a newer intelligent freezing technology. So what that means is it's got an optimized cooling engine inside. It is able to actually freeze to lower temperatures than some of our other instruments. And we can actually measure these more complex samples with components such as these higher viscous excipients such as PEG. And then with regards to the mRNA, we do actually have some customers that are working with oligos and our osmometers. And they're just kind of looking to verify the concentration of the solutions that are carrying their drug product. So a couple of common use cases would be looking at kind of your buffer QC to preserve, protect your critical quality attributes of that product that's being developed. And the second one really is probably a common use case, again, is that compendial release test needed to just confirm the identity and concentration of that oligo product for the safe use. So that would be my answer to that question. I'm not sure if we have questions for the other two guest speakers.
We do have some further questions here. Adrian, would you like to go first with what might the outcome be for different serotypes, for example, AAV2 or AAV3, etc.?
Hey, yes, sure. So for this study, the one I presented, we choose AAV1 because it's mainly 50% intracellular, 50% extracellular at the time of harvest for our process. So this is why we choose AAV1, because like it was the perfect candidate to do this type of study. But I would expect to see a similar trend with another serotype. And it could be very interesting to have a look at AAV2, for example, that is mainly intracellular, and see if we could achieve one of those numbers. And maybe as well having a look at another AAV like 8 that is mainly extracellular, and see if we could basically try to reach almost everything into the supernatant. But I would expect to see like a similar trend. But of course, we have to test it.
Fantastic. Thank you so much, Adrian. And unfortunately, I'm not sure if Lucia's audio will work. She will try and contribute via text, and I can read out her answers once she's able to do so. But we can get started with some further questions that we have received in the meantime. I have one for both Adrian and Lucia. So Adrian, if you're able to take this, what are your current pain points in the use of lentivirus, and how can Osmolality help your process decisions?
So I'm not using lentivirus, to be honest. I'm mainly focusing on AAV. And so it's not the same system, but what, in my case, for AAV, it could be a critical process parameter, like pH, let's say. It could be something we could control in order to maximize our production, but as well be like part of, I don't know, we can think about like a CQA or something like that in the future. But for lentivirus, I'm sorry, like I cannot answer that.
No problem, Adrian. Thank you very much for that. And I have Lucia's response now to the previous question. which was how laborious is osmolality testing of the vectors? And she responds, the testing procedure is very simple and requires minimal sample volumes and timing. To test one standard solution and one sample, it takes around 15 minutes. The training of new operators requires around 1 hour, and so far we have encountered no issues raised by other users on performing osmolality testing with our osmometer. So thank you very much, Lucia, for that. Kendall, I have another question here, which may be our final question, and perhaps applicable to both you and Adrian again. The integral data integrity / management features of the osmometer were mentioned. Can the panel discuss the requirements for data integrity in their workflows? Is there any concern regarding data integrity?
If I start this off from advanced instruments point of view, so we know that data integrity is becoming a real discussion point now with customers and laboratories. And regulations, like I said in the presentation, are ever changing. So we want our instruments to be able to keep up with these regulations, always keeping in line with the Alcoa principles, and these new developing instruments that are coming out, specifically as well with this XT and some software upgrades that we've done for previous instruments, are really keeping the osmometers in line with these ever-changing regulations and that data integrity that customers are really looking for. So I don't know if Adrian wants to take it from the lab point of view.
Me, what I will see to be really beneficial in the future is what we discussed before, like to have like a new instrument inline, online, and currently like it's basically like using an external probe. So we can achieve data integrity, but of course there is room for improvement.
Fantastic, that's great. Thank you so much, Kendall and Adrian, and thank you, Lucia, again for participating via text. I believe that is all the questions that we have received today, so that brings this panel session to a close. Thank you very much for your questions, and once again, thank you to Kendall, Adrian and Lucia for this excellent workshop. This webinar is part of the run-up to our Cell UK in-person conference, part of the Cell Series UK, which will be taking place on the 28th to 29th of October 2021 in London. If you would like further information on the conference or would like a copy of the conference programme, please contact the marketing department or visit the conference website. On behalf of Oxford Global, I'd like to thank everybody for joining us again, and I'd like to say a big thank you once again to our speakers who have put a lot of time and effort into making sure this webinar was as interesting and valuable as possible. All that remains to be said is to have a great day, and we look forward to welcoming you to one of our future events. Thank you.